Well, anyhow, guys, I've got about 1 minute past 9, so I'm going to get started. And so thanks for tuning in. This is the Integrative Aspects of Diabetes, Obesity, Metabolism Research Group, so hopefully you're in the right Zoom meeting. Couple of things to be aware of. Number one, mute your mic if you're not talking, so everybody comes into the meeting already muted. If you want to interrupt the speaker and ask a question, that's just fine. I'm the speaker, so just unmute, get my attention somehow. We ask that you introduce yourself first and then ask your question. Okay, so let's just get started. Whoops, and there we go. Okay, so my name is Dan Beard. I'm the presenter this morning. I'm going to be talking about the link between metabolic energy supply and mechanical power output in the heart. So we have a particular interest in heart failure and how all those things work in heart failure. So what I'm trying to show here is that a lot of the prevalent chronic diseases lead to heart failure. Okay, so this is not necessarily an exhaustive list. It's just an attempt to capture some of the big risk factors for developing heart failure. So you can see diabetes on this list, for example. About a quarter of people in the US over 60 are diagnosed with diabetes. So to me, that's kind of mind-boggling. That's such a big number, and the real number is probably even higher. There's one risk factor that everybody has, aging. And if you look at the prevalence of cardiovascular disease with aging, you can see this clear expected trend. Okay, so I'm going to actually talk a little bit about aging and show you some of the comparisons between the heart failure phenotype and the healthy aging phenotype as we go through. So heart failure, what's heart failure? Heart failure is a little bit like obscenity, okay? And that means that in that we don't really have a good definition for it, but we know it when we see it. So these definitions are from NHLBI and from the American Heart Association here. Getting phone calls. And these are particularly bad definitions because what they base the definition of heart failure on this concept that the heart doesn't pump enough blood to meet the body's needs. Okay, the heart muscle is unable to pump enough blood to meet the body's needs for blood and oxygen. So those are bad definitions because if you look at actual heart failure patients, at least ones that aren't in the not yet at the very ends of their lives, by and large they have totally normal cardiac output. Okay, so these definitions aren't useful. This definition here from this UK organization is a little bit better. It says that heart failure is a condition in which your heart does not pump blood as efficiently around your body as it should. It's not a perfect definition because it begs the question, what does efficiency mean? How do we measure efficiency? How low is not efficiently enough? How high would be too efficient, maybe? There's a better definition I found in this textbook, the Lilly textbook of pathophysiology of heart disease. These definitions really matter, and they're going to matter in this talk, so it's worth kind of getting it straight. So heart failure is present when the heart is unable to pump blood forward at a sufficient rate to meet the metabolic demands of the body. Okay, same kind of definition we already heard, but this or is really important. Or is able to do so only if cardiac filling pressures are abnormally high. Okay, so typically in heart failure, you have some kind of compensation where preload is elevated, and that elevated preload, which has its own consequences, is compensating for helping the heart to deliver cardiac output. So I'm actually adding another or to that definition, which I think is really important. Or is unable to recruit reserve cardiac output to meet the demands of the periphery and exercise. And that's kind of important because that's really the clinical presentation of heart failure. It's inability to exercise, it's shortness of breath, it's inability to walk around the block, inability to walk up a flight of stairs. So it's this ability to recruit pumping power, or which is what we're going to look at, and we're going to try to relate that to the metabolic phenotype of heart failure. So what's the number one predictor of mortality in heart failure? Okay, it turns out that it is the peak cardiac power output. Okay, so that's how much mechanical power the heart can do in voluntary exercise. All right, so this is not necessarily a useful clinical metric, and because in order to measure cardiac power output, you have to measure stroke work or cardiac output during voluntary exercise. So it's not that easy to assess, but this predictor does give us some insight into the mechanisms linking metabolic power supply and mechanical power output. Okay. So as a matter of fact, another good prognostic is a metric of metabolic energy state, the ratio of phosphocreatine to ATP in the myocardium. So these data here on the left come from a meta-analysis that we did looking at myocardial creatine phosphate ATP ratio in human hearts, where the normal are in blue, diseased are in red, and this is that measurement as a function of heart rate. The measurement, as you can see, is highly variable. It varies a lot from lab to lab, but if you look at the measurements within one lab, and these measurements are from the Oxford, Stefan Neubauer group at Oxford, you can find a cutoff value for creatine phosphate ATP ratio that does a very good job of separating mortality rates in terms of these mortality curves, days of follow-up after measurement of phosphocreatine ATP ratio, with looking at subjects with phosphocreatine ratio, ATP ratio less than or greater than 1.6. So again, this is prognostic is not really clinically useful because there are relatively few places where this sort of measurement can actually be done. So it can't be done here, for example. But the deeper question is, what's behind this phenomenon? So here's a framework for thinking about these things. So the heart oxidizes fuel, okay, food, carbohydrates, fats, a little bit of ketones, to generate chemical energy in the form of ATP hydrolysis potential, and then it transduces that ATP hydrolysis potential to do mechanical work. And I like to go through these numbers when I talk about these concepts, because it's useful to be able to have an intuition for what are the numbers associated with how much energy the human body dissipates. So at rest, typical human body is dissipating energy at a rate of about 100 watts. Okay, so that's like an old-fashioned light bulb, right? And so you're burning, sitting in a chair, not doing exercise, you're burning energy at about that rate. About 5% of that energy, or 5 watts, is being consumed by your heart, the myocardium. Okay. There's about a 50% efficiency of converting the chemical energy in primary carbon substrates to ATP hydrolysis potential. So myocardium is burning ATP or hydrolyzing ATP at a rate of about 2.5 watts. And the power output of the heart is about 1 watt. Okay, so is that a lot? Is that a little? It's actually kind of a small number. If you think about a diesel engine, for example, a diesel engine puts out, many hundreds of thousands of watts of power. Okay, so so by the way, what's the thermodynamic efficiency here? The thermodynamic efficiency of converting this 5 watts of food energy into 1 watt of power is about 20%. So a good diesel engine, by the way, is about 50% efficient. So that's actually much better than the heart. Actually, the diesel engine is a little bit easier to understand than the heart, in my opinion. For those of you who might have some engineering backgrounds, it's worth pointing out that a diesel engine or a gas engine is kind of roughly doing the same thing as the heart. It's oxidizing fuels to transduce chemical energy to do mechanical work, right? The mechanics of the process of the diesel cycle or the Carnot cycle are understood in terms of the pressure volume loop, just like, almost just like the pressure volume loop for the heart. Okay, but anyhow, it's a digression. Back to the heart. So what I'm suggesting, and what we want to sort of try to understand, is how this phenomenon, okay, of mechanical power output and differences in mechanical power output in failing versus normal hearts, or healthy versus diseased hearts, is related to this energetic phenotype. Okay, it makes sense that these things are related: metabolic power supply and mechanical power output. But the question is how, right? Mechanistically, how? And I do want to bring up a common misapprehension, which I think is important. So sometimes data like these that we're showing on the left are interpreted to mean that there's some kind of a mitochondrial dysfunction, and that mitochondrial ATP supply and myocardial ATP use are out of balance somehow in heart failure. And so you can find figures like this figure here in all over the place in review papers. So in my opinion, this is a really, really bad way to think about things. Okay, so first off, energy demand tends to be elevated in heart failure. Okay, not reduced. But ATP supply and demand are always in balance. And they're always in balance because the myocardium turns over all of its ATP pools, every few seconds, maybe every 10 seconds or even faster than that. So ATP is constantly being hydrolyzed and resynthesized. And if we're ever out of balance for more than a few seconds, you would die. Okay, so those things have to be in balance all the time. ATP demand is elevated in heart failure, and so therefore ATP supply is elevated in heart failure. Okay, so why is ATP demand higher than normal in heart failure? So think about some of the things that lead to heart failure. Okay, so for example, hypertension and stenosis. These things require the heart to do extra work to eject the blood, right? Obesity requires an elevated cardiac output, okay? So moreover, the sort of remodeling that happens in heart failure, namely dilation, that increases the needed tension development in the myocardium, requiring increased ATP use. So when ATP hydrolysis is actually measured, and it's hard to see, so you might just have to believe me, but on this graph, we're looking at ATP hydrolysis rates in normal versus reduced ejection fraction heart failure and aortic stenosis, and ATP hydrolysis rates are elevated in both cases. So just to hammer home that point, ATP supply and demand are necessarily in balance in the myocardium. ATP demand tends to be increased in heart failure compared to in a healthy condition. So when we actually purify mitochondria from failing human hearts, these are measured oxidative capacities for ATP synthesis. On PM is pyruvate malate substrate, and PC is palmitoyl carnitine. So we find that the failing hearts, red, versus the age-matched controls, blue, are show a slightly reduced but not statistically significant reduction in oxidative capacity. When other people have made the same kind of measurements, they actually show in the failing hearts, so heart failures, for example, on pyruvate malate, this is from the Dale Hamilton group, their measurements show that mitochondria have a greatly enhanced capacity to synthesize ATP in heart failure. So, so I'm not sure what to make of these results, but I think it's safe to conclude that there's no clear evidence that mitochondrial impairment in heart failure. Okay, so we're going to go back in time for a minute. So one way to get a handle on how mitochondria work in vivo is to assay how they work in vitro. Okay, so these are respirometry data from guinea pig liver mitochondria. Okay, and these experiments were done in the Britton Chance lab in the 1950s. So these kind of data can be used to make a simple model that relates oxygen consumption to ADP concentration. So ADP is obviously primary substrate for ATP synthesis, right? It's ADP that gets phosphorylated to make ATP and oxidative phosphorylation. Okay. So if you realize that oxygen consumption is stoichiometrically related to ATP synthesis, so from this kind of simple relationship, you have a computer or mathematical model that can be used to simulate mitochondrial ATP synthesis in vivo. Okay, and so that's just what this same group did 30 years later. So these are a couple of references from Britton Chance group from the 1980s, and these are some of the first measurements of phosphate metabolites in human skeletal muscle in vivo. And essentially what these guys were doing is we're putting their arms inside relatively narrow bore high field magnets so that they could spectroscopically assay phosphate, ATP, phosphocreatine. Okay. And here is the simple model that was first elucidated for the guinea pig liver mitochondria right here. Okay, so this is that sort of Michaelis-Menten kind of relationship between, flux and substrate concentration. So the substrate is ADP. So with the simple kind of equilibrium assumption, you can relate the ADP concentration to ratio of phosphate to phosphocreatine. Okay, and hallelujah, look at that. The relationship observed from the guinea pig livers in 1955 matches observations on the human arm energetics in 1985, 30 years later. Okay, so here's a more recent example of the same kind of thing. It's still 20 years old. The experiment here is to occlude blood flow with a cuff. Okay, and so phosphocreatine goes down, and inorganic phosphate goes up when the blood is occluded. Okay, and when the cuff is released, you get a recovery. And this simple sort of model explains what's going on. Okay. Now, okay, so let's just stay on this slide for a second. So for some strange reason, nobody ever did this kind of a thing for heart muscle. Okay. So actually, the reason nobody ever applied the same kind of thinking to the heart is that around the same time that Brad Chance was putting his arm in a magnet, the cardiac energetic field adopted some, what I think are some very strange ideas that held the field back for a couple of decades. But actually, it turns out that you can do the same sort of in vitro to in vitro kind of thinking for the heart. And so these data are from experiments that we did a while back, looking at some of the state variables of the electron transport chain as a function of oxygen consumption rate on the x-axis here. And so the way we do these experiments is that we titrate in ATP hydrolyzing enzymes, and we measure NADH and cytochrome C redox, ADP and membrane potential as functions of oxygen consumption rate or of ATP demand rate equivalently at different levels of inorganic phosphate concentration. Okay, so the red and the blue lines are model simulations. And so we can use these kind of data to identify models representing, mitochondrial energy metabolism. So, here's a diagram of a model. The details of the model aren't important for what I'm going to show you. You can see that we're using models that are much more complicated than that simple Gale-Minton kind of equation that we saw applied to skeletal muscle. And we've been working on these models for more than a decade, and so they're growing in detail. But the important thing, for our purposes here is to know that we can use them to simulate in vivo energetics, just like Chance was doing in the 1980s for skeletal muscle. Okay, so we use these models to simulate of mitochondrial energetics to simulate energetics in the myocardium by coupling the mitochondrial model to a cell, and in the cell we account for a few reactions in the cytosol. So ATP hydrolysis, so just demand for ATP, and then the creatine kinase reaction, the adenylate kinase reaction, and we can simulate in vivo energetics by varying the ATP demand rate to compare model predictions to measure to measurements of the creatine phosphate to ATP ratio and the inorganic phosphate concentration. Okay, as functions of myocardial oxygen consumption. So that's what I'm showing up here. So these experimental measurements, the data points here are made in a canine myocardium in vivo. And, so in these experiments, the animals anesthetized, and so changes in oxygen demand or ATP demand are driven by pacing the animal and also by administration of catecholamines. All the data here, by the way, come from Jay Zhang, who was at the University of Minnesota at the time. And so we also convinced Jay to do a transient experiment. In this case, it was done in a pig, where what happened here is that the LAD, the left anterior descending coronary artery, was occluded. When the occlusion happens, creatine phosphate goes down, inorganic phosphate goes up. When the occlusion is released, you get a recovery. So for all of these experiments, and you can see that the model predictions go through the data really nicely. Okay, so these are all, healthy, normal control animals. All right. The other thing that's important to point out is that for all of these experiments, so, these are steady state experiments in these two plots on the left. There's a transient experiment on the right here. The model, computer model for mitochondrial energetics was identified from data obtained from mitochondria from rat myocardium, right? And now we're using the rat mitochondrial model to explain dog and pig energetics. So, take that with a grain of salt. So remember, these kind of simulations started actually using models identified from guinea pig liver mitochondria to analyze human skeletal muscle. So hopefully it's not such a great big leap. But I guess, can we make this leap to humans is really the next question. And what about, can we use this kind of models to understand disease? So before we start talking about humans, let's look at disease in a dog model of heart failure. So our collaborators from University of Minnesota, this is Jay Zhang and Bob Bakke, had a dog model of heart failure. It was an aortic constriction pressure overload model. And the model captures, so this is the same control data I just showed you, the normal case. The model captures some of the phenotype you see in humans in heart failure, namely a reduction in the creatine phosphate to ATP ratio. So Bob and Jay looked at individual animals from this model, and they measured myocardial energetics at different work rates. So here's normal. Here's an animal with hypertrophy, but not yet decompensation. And here's a more severe case. In this severe case, the animal has heart failure. It's got an end diastolic pressure of more than 20 millimeters of mercury. And you can see this really clear reduction in creatine phosphate to ATP ratios. Okay. You can also see that the model matches the data really well. Okay. It matches the disease data just as well as it matches the control data. So how does it do that? So we actually had measurements of myocardial metabolites from these animals. And the important thing to account for in making these model predictions is to match these data is that the pools of total creatine and total adenine nucleotides get depleted in hypertrophy and in decompensation. Okay, so total creatine is the unphosphorylated plus the phosphorylated creatine. Total adenine nucleotide, the big contributors to the total adenine nucleotide are the ATP, ADP, and AMP. Okay, and so these pools become progressively depleted during hypertrophy and decompensation, and we can adjust the model to match the measured changes in these pools. Okay, and we get predictions that go right through the data really nicely. Okay. So, in fact, the only changes that we made to the control models, we haven't changed the mitochondria at all, is we've just changed these cytosolic metabolite pools. Okay, so to briefly summarize what, get a box in the way here too. So what we've seen so far is that, like I said, ATP supply and demand are in balance in the myocardium. ATP demand might be elevated in heart failure compared to the normal case. The mitochondria from failing hearts from humans show little or maybe no impairment in oxidative capacity. And when we do these simulations of energetics in animal models, large animal models of heart failure, the models match the data perfectly well, as long as we account for these changes in these cytoplasmic metabolite pools. So I'm going to make a claim that we have a pretty good understanding of how energetics is regulated in vivo in normal physiology, and at least part of what's driving the heart failure phenotype.
Dan?
Capture what's measured. Yeah, go ahead.
I just have a quick question. This is Bonnie. So I know that I think, if I remember correctly, you had said that in these canine models they had an LAD occlusion, and is that what caused the heart failure?
Good question. No, that was a different. That was a transient LAD occlusion that we were looking at before. So no, in this case, this is an aortic constriction pressure overload model.
Oh, okay. I was wondering what kind of hypertrophy there was, and if it mattered, what these pools would look like then. Okay, thank you.
Yep, sure thing. Yeah, I should have made that clear. Thanks for the interruption. Okay. So where were we? So I just want to point out that this depletion of metabolite pools is actually a pretty old story. Okay. First reported in the 1930s. So these are measurements on creatine in failing hearts, right? Normal versus failing. Okay. And here we have more measurements on total creatine, but we can also have reported in these papers were creatine phosphate levels. Okay, so these measurements, this phenomenon has been appreciated, or at least observed, for a long time, but very little attention has actually been paid to it. And I actually think it's crucially important to the metabolic and the mechanical phenotype of the failing heart. Okay, so I've shown you a lot of simulations on animals. Let's switch gears to humans. In collaboration with Jordi Yakovlevich at Newcastle University, we built an age-structured population model of human myocardial energetics. And so these are the data that we have from Jordy. So all of the measurements that we have, and so the model reflects women, and that's what I'm trying to indicate in my my representation of the virtual population here. Okay, so what we did in order to kind of understand what's happening with aging in humans, we postulated that the metabolite pools that are changing in heart failure are also changing in aging. Okay, and so we created a statistical model to represent the probability distributions of those pools, the adenine nucleotide, the creatine phosphate pools, okay, and also a covariance among those pools as functions of age, okay, and we identified the population model by matching model predictions to these data here. Okay, so the red dots represent measurements made on individuals under resting conditions. Okay, and the solid line is the median model prediction, and the gray boxes, the dark gray areas are the 50% confidence range of the population, and the light gray boxes are the 90% confidence ranges. So the median populations, the median predictions go through the medians. The 50% confidence ranges capture 50% of the data, and the 90% confidence range captures 90% of the data. So that's how the model's parameterized. Okay, so this is all identification of the model. Right? And inorganic phosphate's not directly measured, but we predict that inorganic phosphate concentrations are gradually going up with aging, and that's actually kind of important. It's important if it's correct, and we'd like to see if somebody can measure that and see if our model predictions are correct there. But so what can we do with these kind of things? So one thing we can do, and this I think is really, really cool, is we can predict maximal cardiac mechanical power output by computing the maximal metabolic power supply. Okay, so in this case, we don't have a model of the mechanics. Okay, so we're making a really big assumption, and the assumption is that the efficiency of the cross bridges is constant. Okay, but given that assumption, what we find out is that if we simulate now, again, these data were all that the model was identified from these data, which were all at rest, and now we make a prediction of how much mechanical power output the heart can produce. And what we're plotting here are the individual data on the left, and just means and standard deviations from the same data set on the right. Model predictions are the same. So the model prediction kind of nicely captures this trend in the data that with aging, mechanical power output goes down. Maximal mechanical power output goes down. Okay, and it's driven entirely by this reduction in these cytoplasmic metabolite pools that we're predicting occur with aging and impair the myocardial capacity to synthesize ATP at physiological free energy levels, and so the resulting changes to the energetic status are predicted to play a causal role in contributing to reductions in maximal cardiac power output with aging. Okay, so again, like I said, we made this huge assumption about the relationship between the mechanics and the metabolic power supply in those simulations. But we've also started looking at trying to understand the mechanical part of the problem using computer models. And we've built a computer model of muscle dynamics and cross-bridge kinetics that is sensitive to metabolite changes. Okay, so this is the typical kind of cross-bridge cycle, but the cross-bridge cycle, kinetics of the cross-bridge cycle and the dynamics of the cross-bridge cycle depend on binding and unbinding of phosphate metabolites, right? So we built that model using data on elastic and viscous moduli at different, perturbation frequencies and different phosphate ADP concentrations. So this is some of the data we used to build the model. And what we can do with the model is now we can predict things like sliding velocities at different ATP concentrations, and we can predict force redevelopment, sort of the slack restretch KTR experiment that muscle people like to do at different inorganic phosphate concentrations. And the model captures the effect of the metabolites on tension development, sliding velocities, rates of tension development, and magnitude of tension development. So the model captures the link between the metabolic state or the energetic state and the mechanic state. And so what we can do is we can put all of these pieces together, and this is a model of a rat. Okay, so I'm showing this rat picture here. So specifically, we can take our cross-bridge model and our mitochondrial model, and we can put that in a model of a cardiomyocyte, which has a calcium, which generates a calcium transient. We could take that cardiomyocyte model and we put it into a model of the heart. Okay, kind of cool. And then we can put that model of the heart into a really simple circulatory model. So this is a really crude circulatory model we can get with some lumped elements representing resistances and compliances of the pulmonary circuit and the systemic circuit. And we could get as complicated and sophisticated as we want here. But what's important, what's really cool about this first ever simulation we did a few years ago of coupled mechanics and energetics was, if we take the normal metabolic model, that is, now we're doing this kind of combining all these species together. We're taking the normal metabolite pools from the healthy dog, and we have a rat model of mitochondria and a rat model of cross-bridge and a rat model of the heart and the circulatory system. And we can predict, for example, a normal pressure volume loop, right? If we then impose the failing energetics, by that I mean we just sort of turn a switch on only the myocardial metabolite pools, what happens is we get heart failure. Okay, and this is all model predictions here, right? So heart failure, I mean, we get a reduced ejection fraction, we get elevated, we get congestion, right? We get this really high filling pressure, which is needed to produce even a slightly reduced stroke volume or slightly reduced cardiac output. That's the red curves. The green simulations, what we're doing here is we're actually simulating what happens if you have a drug which can enhance the kinetics of the cross bridge. And it turns out if you can counteract the metabolic defect by stimulating the cross bridge to go a little bit faster here. Okay. So Rachel Lopez is a graduate student in the group who's using a pressure overload rat model of cardiac hypertrophy to dig a little bit deeper into the hypotheses that are generated by this computer model, and also to interrogate upstream mechanisms of purine nucleotide regulation. And there's Rachel there. Okay. And so Rachel and also Bahadur Marsban, who's a postdoc in the group, have developed this workflow for taking a lot of measured data, things that we can measure in the lab and things that we can measure with the help of people in the cardiovascular phenotyping core, things like all of these metabolite pools in the myocardium, the mitochondrial oxidative capacity, parameters associated with the cardiac geometry, okay, and also things we can measure like stroke volumes and cardiac output, things that we can measure from echocardiography. And the workflow is to put all these things into that same model, and then to make predictions about the energetics, and then to also make predictions about how specific components of the energetic phenotype actually cause the mechanical phenotype in the rat model of heart failure. Okay, so I'm not going to tell you about any more about Rachel's stuff. You can ask Rachel, maybe invite her to give one of these seminars. Rachel's, or you can wait for the paper, because at this very moment, Rachel's probably writing this paper. What I want to tell you about is something a little bit more preliminary and kind of fun though. I'm going to take that rat. What I did is some simulations where I took that rat cross-bridge model in this rat cardiovascular systems model, and I turned it into a human model. Okay, and I did that in a kind of crude way. So, like I said, it's a little preliminary, but really what I did is I just slowed down the cross-bridge kinetics, and I slowed it down so that we're getting an ATP hydrolysis rate or cross-bridge turnover rate that matches what you expect to get from a large animal like a human. Okay, so cross-bridge turns over a little bit more slowly in large animals than it does in small animals, and of course heart rates are smaller also. Okay, and we can simulate cardiovascular mechanics in humans. And what I want to do, so here's a simulated cardiac cycle, left ventricular pressure, aortic pressure, here's a pressure volume loop. I want to do these kind of simulations, and I want to couple them to the myocardial energetics. Now I want to look at this relationship between peak cardiac power output and age. in our age-structured model. Okay, so so the way I did that is I said, okay, first of all, we're going to simulate the normal 20-year-old. Okay, the average young adult female human. All right, and putting it, putting all of these pieces together. Let me just show you all these plots. Okay, so we can simulate what happens to, so what happens during systole is that there's this transition from this non-permissive to this permissive state in the cross-bridge cycle. So this is the fraction that's not in the non-permissive state, or the fraction that's calcium bound. It goes up and it goes down in diastole. The fraction that's attached is a little bit lower. The sarcomeres shorten, okay? And then during filling, they stretch back out again. We predict the pressure volume loops. Here's that cardiac cycle. Let's take the time here. And this is kind of a neat thing. We can predict the rate of detachment of the cross bridge. And so from this rate of detachment, we can integrate under that curve, and we know the cycle time, the cycle time, the average cycle time, or the cycle rate, okay? And so we're able to make a prediction of how fast cross bridges are turning over in the human myocardium. All right, we put all that together. And so for the resting baseline simulation, we have a cardiac output of 5 liters per minute. I set it to be that. I thought, well, that's a nice round number. And with an ejection fraction of 60%, and we have a power output of 1.2 watts, okay? The cell model then predicts what is the ATP hydrolysis rate associated with that resting state. And then the energetics model then tells us what is the resting ATP, ADP, and inorganic phosphate in the myocardium. Okay? So that's rest. So what I want to do is I want to make the model exercise. Okay? And the way I make the model exercise is I push the heart by increasing the heart rate. I have to then dilate the periphery, okay, just as if you're exercising, right? I increase systolic calcium. I decrease diastolic calcium, and another thing you have to do is you have to constrict the veins in order to keep the preload up. So I'm gonna make all of these changes in a way that maximizes cardiac output. Okay, and when I do that, and by the way, for this first simulation, I'm not coupling the energetics and the mechanics. So I'm assuming that the energetics stays fixed at its resting baseline state. So I'm not doing the energetic simulation. Like, how much can I push this system? And it turns out I can push it like crazy. I can get these really, really big ejection fractions, 88%, and I can get power output of 7.5 watts and cardiac outputs of really high cardiac outputs. Okay, so this would be actually, well, I'll show you. I'll compare this to the data in a minute, but I feel this is higher than normal. This is getting into sort of trained athlete kind of regime for maximum cardiac output. But under these conditions, the ATP hydrolysis rate is higher. So, back for the resting case, it was about 0.5 millimolar per second. So it's about 5 times higher. Okay. And at this high ATP hydrolysis rate, ATP is maintained pretty well constant, but inorganic phosphate levels are really, really high. Okay. And it turns out that if we now couple the energetics and the mechanics model together, this really high inorganic phosphate level is going to impede systolic function. Okay, and it's going to make it so that we can't achieve these kind of cardiac outputs. So that's what I'm showing here. So if we put the mechanics and the metabolism together, this is the maximum cardiac power output I can get. This is the maximum cardiac output. This is the maximum ejection fraction I can get. So in this simulations, we can see we're getting an elevation of systolic pressures. The pulse pressure may be a little bit unphysiological. I'm not sure. The fraction of the cardiac cycle, by the way, this is not time in seconds. This is fraction of cardiac cycle. The fraction of cardiac cycle, which is systole, is getting bigger and bigger. So there's less and less time for diastole. That's what's happening physiologically. The model gets a little glitchy. Okay, like I told you, it's preliminary, but we have a lower ATP hydrolysis rate, and we have a maximum inorganic phosphate at maximum exercise of about 1.5 millimolar. Okay, so just to summarize what we're doing, so here's the resting conditions. if we exercise it, but we don't assume that there's any change in the energetics from the resting state, we can get kind of ridiculous pressure volume loops and really high ejection fractions. But if we couple the energetics and together, this is what we get. All right. And that's really, really, really interesting. And so let me just show you what I'm doing. So how did I get, so this is my resting state here. Okay. This is now cardiac output, ejection fraction, power output, and inorganic phosphate. That's functions of how much I'm turning up the heart rate. Right. And so when I turn up the heart rate to about 180, okay, that's where I get the maximum in all of these outputs, okay? And if I try to go to higher heart rates, ejection fraction starts going down, power output starts going down, and so we've gotten kind of, and I'm not showing all these simulations in between, that's why there's this big dash line. These simulations are kind of expensive. You know, when we get to this sort of saturating maximum of predicted inorganic phosphate. So what I'm predicting here, the model actually as an emergent property for a normal 20-year-old actually predicts maximum heart rate, which is right about physiological maximum heart rate, and it predicts maximum cardiac output and maximum power output, okay? And we can look at that peak cardiac power output against data, and we're using the median average 20-year-old energetics, and we're getting the median average 20-year-old peak cardiac power output. So we're predicting this point on this plot really, really well. And this is sort of an emergent property of putting these things together. And the limitation on the cardiac power output is the mechanical power output is the metabolic capacity to produce to deliver ATP. Okay. All right. What about the 80-year-old? Now the 80-year-old simulations are a little bit messier. Okay, and I'm not changing anything in the model about the cardiovascular system. I'm not changing the geometry of the heart at all. All I'm doing is now I'm taking the metabolism in the myocardium and I'm making it equivalent to the average median 80-year-old from our population model. And so under resting conditions, what you get is you get a little bit of a reduction in cardiac output, a reduction in power output, and we get an ejection fraction which is lower than normal, so that's not correct. You know, in other words, what we get is we get heart failure with reduced ejection fraction just by making the metabolism of an 80-year-old. Okay, so that's not necessarily correct for healthy aging, and we can predict this elevated inorganic phosphate. And because the cardiac output and the power output are lower, the ATP hydrolysis rate is a little bit lower than it is for the 20-year-old. The 20-year-old it was about 0.5 millimolars per second. Okay, and so then let's try to exercise that 80-year-old. And what happens when we exercise the 80-year-old model is we hit this maximum heart rate at a much lower heart rate. And look at that, we get maximum heart rate of about 140 for the 80-year-old. And then after 140, you get diminishing returns on trying to increase heart rate even beyond that. So we can predict age-dependent differences in cardiac power output. We can predict age-dependent differences in maximum heart rate just by putting these metabolism and the mechanics together. So that's kind of cool. And where do we land on this plot? So actually, the model is doing worse than the regression line, okay, in terms of peak cardiac power output. The model prediction in terms of peak cardiac power output is coming pretty close to the two individuals that are measured, but it's actually, you know, kind of low here. And I think it's kind of low maybe because we're not accounting for any other changes, right? We're not accounting for any other compensations, okay? And so what I'm showing you now, just to be clear, is a virtual population, but it has exactly two members of the virtual population, right? The median 20-year-old and the median 80-year-old. So we have some work to do to really look at this, you know, in a little bit more depth. But to summarize, this what I think we conclude from these simulations. So what's happening to cardiac energetics and heart failure? Okay, so there's just depletion of these metabolite pools, It's a major driver of the energetic dysfunction that we see in heart failure, okay, in animal models and in humans. The mitochondrial oxidative capacity, it might be reduced or it might be unchanged. It's not too clear. It looks like it clearly, mitochondrial density and mitochondrial ATP synthesis capacity is going down in the rat models. And that seems clear, and it may be that it goes down in the humans as well. We just don't have enough data to see statistical significance. But again, we think one of the big drivers is the metabolite pool changes. Depletion of these metabolite pools, we think, is also a driver of the energetic phenotype in the aging heart. And we put the mechanics and the metabolism together. It turns out that energetic status is a major determinant of the maximum cardiac output under healthy conditions and also in healthy aging. And there's a corollary to all this, which is that if we can restore the metabolite pools, then that's going to lead to, the prediction is that's going to lead to improved mechanical function in both heart failure and in aging. Okay, so I think that's all I've got. Yeah, so here's just a list of the people that are current lab members and collaborators at the University of Michigan, and also some of the past people who worked on some of the simulations I showed you. So a lot of I just want to point out a couple of names. Jason Basil, who's now at Michigan State University, assistant professor, did a lot of the work on a current mitochondrial model. Shavendra Tiwari worked on the Crossbridge model. Who else do I want to point out? It's some of our collaborators at other universities. So we're working with Paul, Edith Jones, in particular, is working with Paul Jansen at Ohio State to try to do some mechanics of in human cardiomyocytes, so that we can actually maybe build a little bit more credible human model rather than just trying to scale the rat model. Jeroen Jensen at Academic Medical Center and Jordi Akolovic are helping us make energetic measurements in human myocardium in vivo. We have a new project that we're just launching with Naomi Chesler at University of Wisconsin, who's sort of really interested in looking at all of the things I just showed you, but on the right side, the right ventricle and the pulmonary circulation, which is kind of maybe a neglected half of the heart. And here's a picture of the group. Obviously taken before pandemic days. I wish we could all get back together, and I'm hoping we can all get back together soon. Thank you very much. And we got time for any questions. Thanks a lot. So just a reminder, I think I can, maybe it matters, I don't know if it matters to stop sharing. Oh, I have a question in the chat here from JH, and I don't know who JH is, I'm sorry.
It's Jonathan Herrera.
Oh, Jonathan Herrera. Hey, John, what's happening? Yeah, I sent you an email a while back, by the way. I never heard back from you. So John says, how cardiac energetics change in patients with heart failure that have gone through cardiac rehabilitation? Yeah, like versus not go through training. That's a super question. Unfortunately, I don't have an answer for you. But we'd really like to address that question in future studies. So I guess the question is, another way to look at that is this, if we looked at the one particular aspect of the phenotype which we're zeroing in on, which is adenucleotide and creatine phosphate pools, do they actually change with training? We know they do in skeletal muscle, right? We know a lot more about skeletal muscle than we do about the heart in some ways, and what we know about skeletal muscle often translates to the heart. It's just like what I showed you. So my suspicion is, the answer to your question is, yeah, they'll change, and they'll change in the right way.
Thanks, Dan.
Yeah, no problem. So we're also, by the way, this is something that another thing that Rachel's working on is we're looking at the pathways that are associated with degrading those pools, particularly the purine nucleotides. And we're trying to phenotype some rat models where we've knocked out enzymes associated with those degradation pathways. And preliminarily, it looks like if we knock out those enzymes, we can preserve the nucleotide pools, and we can also then preserve. When we preserve the nucleotide pools, we actually restore, or at least partially restore, mechanical function. And we're also trying to do the same sort of thing with drugs. Okay, I'm not hearing any more questions. If you are asking questions, you can put it in the chat, or you can speak up. But don't forget to unmute yourself if you're going to speak up. Okay. All right. So I'm not hearing anything, Sarah. So I think we can wrap it up. Feel free to track me down if anybody has any questions or comments.
Thank you, Dan.
Yep. Thanks so much for having me.